Welcome to ENCREASY, which means approval in Greek, and was founded on the premise that biotechnology, medical devices, and pharmaceuticals hold the promise of a better future for everyone. Making good on that promise requires innovative approaches, world-class science, regulatory know-how, and a team dedicated to bringing all of it together. The journey of a drug from laboratory conception to market shelves is a complex process, intricately navigated through the Common Technical Document, or CTD. The CTD is not merely a formality, but the backbone of a drug's approval process, serving as a comprehensive dossier for regulatory agencies worldwide. It harmonizes the submission process, allowing for a systematic review of the drug substance and product. Understanding the CTD structure and construct is paramount for any entity aiming to achieve regulatory compliance and market access. In this discussion, we turn our focus to Quality Module 3 of the CTD, a pivotal element in the regulatory submission process for pharmaceuticals. The Quality Module 3 is the cornerstone of a drug's approval pathway, encompassing detailed data on the drug substance and product. The ICHM-4Q guideline, part of the CTD for the registration of pharmaceuticals for human use, outlines the scientific and technical aspects of the quality section. Its criticality stems from its role in assuring regulatory authorities of a product's quality and even safety and efficacy. Understanding and mastering this module is not just a regulatory necessity, it is a strategic imperative for any pharmaceutical or emerging biotech sponsor looking to navigate the complex waters of drug approval successfully. Herein we'll dissect the fundamental nuances of the CTD Quality Module 3, providing strategic insights into how to leverage its sections for a compelling submission. In the pharmaceutical industry, the CTD is the cornerstone of drug regulatory submission. Designed for uniformity, it is the lingua franca between innovators and regulators. But beyond compliance, a well-crafted CTD is a strategic asset, smoothing the path for approval. It is a narrative that, when articulated with precision, can significantly influence a drug's time to market. A drug substance's journey begins with its general information, or section S1, where its identity is established through nomenclature, structure, and general properties. This foundational knowledge assures regulators of the substance's legitimacy and traceability. Detailing the drug substance starts with its nomenclature, where both the non-proprietary and proprietary names are given, along with chemical names and, of course, the structure, whether a small or large molecule. For example, a simple drug like aspirin would be listed with its chemical name acetyl-salicylic acid alongside its structural formula. This sets the stage for regulatory clarity. Next, the manufacture section, S2, delves into the active substance's creation, detailing the manufacturer's information, the description of manufacturing processes, and the stringent controls in place to ensure consistency and quality. This transparency is crucial in building trust with regulatory authorities. Manufacturers must disclose the origins of their drug substance, the regulatory starting materials. Then a good strategy is to provide a comprehensive description of manufacturing that not only states the what, but also the why behind each step, whether it's a series or a convergent synthesis. For a biologic, this might include cell line development, culture conditions, and purification steps, each justified by scientific rationale. Characterization, on the other hand, is the substance's fingerprint, and is where the substance reveals its nature, including the structure and impurities, which are vital for understanding the drug's safety profile. A thorough elucidation of structure and identification of potential impurities in section S3 can preempt regulatory questions. It's where a sponsor might illustrate how impurities are quantified and controlled, like the limits set for benzene in soft drinks, to make a simple example, to ensure patient safety. The Control of Drug Substance section, or S4, is where specifications are outlined, analytical procedures are detailed, and validation of these procedures is confirmed, ensuring the drug substance meets the highest quality standards. Specification setting is both an art and a science. Sponsors should leverage historical data and industry standards to justify their specifications, much like how the pharmacopoeial standards are set for common excipients. Drug specification setting, however, is significantly influenced by manufacturability, reflecting a balance between the ideal clinical performance of the drug and the practical aspects of its production. Here are some key ways in which manufacturability influences drug specifications: production consistency, scalability, quality control, material availability, process-induced variations, stability, regulatory compliance, as well as cost-effectiveness. Reference standards detailed and described in sections S5 and P6 are high-purity substances used as a benchmark in the analytical testing of both the active as well as the final dosage form to ensure consistency and accuracy in results. They serve as a critical tool for validating the identity, strength, quality, and purity of drug substances and products during quality control processes. In Drug Substance Stability section S7, presenting real-time and accelerated stability data can fortify confidence in the drug's shelf life claims. A case in point is the detailed stability studies required for vaccines, for example, ensuring their efficacy even when distributed globally. Stability studies also play a crucial role in supporting the selection and validation of container closure systems, sections S5 and P6 for the active substances, and the final dosage form. These studies help to ensure that the container closure system effectively protects the drug substance throughout its intended shelf life. Here's how stability supports container closure for drug substances: protection against environmental factors, physical integrity, preservation of potency and purity, microbial contamination, and support for shelf life and storage conditions, and tailored for specific formulations. Different drug substances and formulations may require different types of container closure systems. Stability studies help in customizing the choice of the system based on the specific needs of the drug substance. Lastly, stability testing is documented, demonstrating that the active substance will maintain its integrity and efficacy throughout its proposed shelf life. In turning attention to the drug product description and composition section P1 serves as the product's blueprint. Here, the strategic inclusion of excipient compatibility studies, similar to those presented for novel drug-eluting stents, for example, can demonstrate a comprehensive understanding of the product's formulation. The description and composition of the drug product paints a complete picture for the regulatory reviewer, detailing what the product contains and how it is formulated. The Pharmaceutical Development section P2 should tell the story of the product's evolution. A narrative that describes the decision-making process, like the rationale behind the choice of a tablet versus a capsule form for better bioavailability, can be persuasive. The P2 section, focusing on pharmaceutical development, is often considered the lifeblood of a drug application because it provides a comprehensive narrative of the drug's development journey, elucidating the scientific and technical rationale behind the formulation and manufacturing process. This section is crucial, as it not only demonstrates the robustness and thoughtfulness of the development process, but also establishes the groundwork for the drug's quality, safety, and efficacy. By detailing the decision-making process, including choices of excipients, how the dosage form arrived, the manufacturing methods, and control strategies, the P2 section helps regulatory authorities understand the product's intended performance, thereby facilitating a smoother review and approval process. This section intricately details the formulation process, highlighting the rationale behind each decision made during the development phase. It includes discussions on the choice of ingredients, the formulation strategy, and the manufacturing process, and ultimate controls outlined in the drug product manufacturing section P3. By documenting the developmental history and the scientific reasoning for the chosen approach, the P2 section ensures a thorough understanding of the product's design and its intended performance, thereby facilitating a smoother regulatory approval. Controlling excipients in section P4, especially novel ones, requires a balance between innovation and safety. Presenting toxicity studies, as one would for a new preservative in ophthalmic solutions, for example, underscores a commitment to patient safety. Control of excipients is a critical section, especially for novel excipients or those derived from human or animal sources, where justification and stringent control are essential for safety and efficacy. Control of drug product and validation of analytical procedures for the final product must be robust. Using a risk-based approach to method validation, akin to what is detailed in ICH guidelines, can demonstrate a proactive stance in ensuring product quality. The control of drug product section P5 mirrors the substance control section S4, but focuses on the final product's specifications and validation processes, to ensure it meets predefined quality criteria, borrows from the S5 reference standards and justifies the container closures described in P7. Finally, the drug product stability section P8 must be presented in a way that anticipates variability in global distribution. An example is the stress testing done for insulin products, ensuring they withstand temperature fluctuations during shipping. The product's stability is the final act, ensuring the drug product remains safe and effective throughout its intended shelf life, even under various environmental conditions. Navigating the CTD is a strategic endeavor. Each section requires not only data but a story that aligns with regulatory expectations and patient safety. The most successful applications are those that anticipate the regulator's needs and address them proactively. In this process, the principles of good development practices are not just guidelines, but the narrative arc that leads to successful approval, ensuring that a drug's journey from conception to market is both efficient and compliant. The CTD is not just a regulatory requirement, but a narrative of a drug's life story. Each section and subsection serves as a chapter that collectively tells a tale of innovation, safety, and hope. As the pharmaceutical landscape evolves, the CTD remains a steadfast pillar in the regulatory approval process, guiding sponsors through the meticulous journey of drug development. The CTD is not merely a formality, but the backbone of a drug's approval process, serving as a comprehensive dossier for regulatory agencies worldwide. It harmonizes the submission process, allowing for a systematic review of the drug substance and product. Understanding the CTD's structure is paramount for any entity aiming to achieve regulatory compliance and market access. One final note: our company Herein offers a comprehensive service to emerging biotech companies to help them navigate the complex world of product development, including establishing a concise CMC development and regulatory CMC strategy and submission plan. With a splash of augmented intelligence, we're more than a team of experts, we're your partners. Introducing Herein, the leading regulatory CMC consulting group helping organizations comply with drug regulations through a comprehensive drug development model.